Swayam Prabha. Digital India. Educated India.
So that is where this entire thing came, and so you see something that was discovered 20 years back was made use when they had a lot of human genome data, in the sense, okay, I have this data, I should use this. The way to do that is if I can edit that. So that is what these two people did, which is Emmanuel Charpentier and Jennifer Doudna from UC Berkeley, and then I think she was at MPA for some time. So what they did was they wanted to understand whether if I use my CRISPR RNA, as I said, I use this CRISPR RNA, I use a specific sequence, I use this Cas9 enzyme, which is nuclease and helicase, and I put it in a test tube, whether it is able to edit it or not, is what they wanted to try. Initially, any experiment, it doesn't succeed. So this experiment also didn't succeed. They realized that not just the RNA part and the enzyme, there is also one more part that is called as tracer RNA, which is basically this is called as complementary strand. So this tracer RNA, which is complementary to the palindromic sequence. So we thought like palindromic sequence is not important. We only were thinking like these small ones are the ones that are coming from virus. But they realized that this tracer RNA, which is from the palindromic sequence, that binds and that activates this enzyme. So that is important to activate this. You know, enzymes have a specific active site, right? So that is where, so they activate that enzyme, and that gave the complete thing. So once they did that, so they had this nice complex. So this is basically enzyme, which is a helicase and a nuclease, and then this is your RNA, which is complementary to the, in this case, the green one is complementary to your viral DNA, the red one is the palindromic, and this the black one is the palindromic, red one is the complementary to the palindromic, which is activating this. This entire thing is called as CRISPR-Cas9 system. The enzyme is called Cas9, this is called CRISPR. So this is called CRISPR-Cas9 system. This is a tool that is being used to do genome editing. Right? So that is why these are called as molecular scissors. I am calling it scissors because they are actually cutting off your DNA, right? So these are called as new molecular system, these three things. After that, few things happened. I just wanted to show you the structure. This is how the protein structure of your Cas9 looks like. Okay. If you want, I'll just go through the mechanism. But I think I'll just show you a little later. But in addition to that, as you remember, so there is a sequence, and then there is this the complementary to the palindromic sequence. What they did was they have joined these two, and this is called a single guide RNA. So previously you had a separate sequences. One sequence is for your binding to the viral protein, the other is to binding to the palindromic part. Now they have kind of made it together, and that is called a single guide RNA. In the sense, if you have this single guide RNA, depending on these bases, you keep on changing the bases in whatever region you want to edit. You keep changing these bases, it will go there and kind of do the nuclease activity. Let us see a quick example for that. For example, I have this specific, for example, this is a single guide RNA, okay. So this is a nucleotide sequence that should be complementary to the region where you want to bind it. This is your tracer RNA, which is basically complementary to the palindromic part, and this will activate your enzyme. So this will bind to the enzyme Cas9, and this has a nuclease and helicase activity. So this is your, whatever I showed you as a cartoon is this, the molecular scissor. Okay. Now let us see an example how it actually works. Now I have some DNA sequence, some random DNA sequence. I don't know what it is, but it is probably a human genome, part of human genome, and we know that there is this region, green region, which is causing a lot of issue. In the sense, that is what is creating some amino acids, which is, you all know that a lot of diseases are caused because of proteins. For example, Alzheimer's. Alzheimer's is a old age disease where basically there is a protein called amyloid beta 42 that kind of aggregates. That means it forms a lot of plaques and forms. There are very few amino acids in that will cause this aggregation to happen. Now if you can remove those amino acids at a much earlier level, then that protein will not aggregate and will not have the Alzheimer's disease. This is just an example. Same thing for many hereditary diseases, like in principle, autism, right? This is also a kind of a hereditary disease, right? And then you have a lot of other diseases. Even some forms of cancer are also hereditary. So again, all these are because of a certain protein specifics. Now, what are proteins made up of? Proteins are made up of amino acids. So if you can mutate those amino acids, or if you can change those amino acids, they will not be doing causing a negative impact. The same thing will be for positive. If you can enhance specific amino acids, there will be an enhancement in that. So I have a specific genome sequence, I have some gene sequence, and I realize that, now how do I realize that this is the sequence? You have to do a lot of other experiments, and you realize that this is the sequence that is actually causing the problem, and you want to remove it or delete it, right? Or the sequence after this is the region that I want to delete it. Now I design so this is the DNA. This DNA will base pair with its complementary. As I told you, all the DNAs will base pair with its complementary bases. Now it will base pair with that. Now you design your single guide RNA with so this all will be constant because your palindromic sequence is constant. Your tracer RNA, which is complementary to the palindromic, is constant, and this is part of the region which activates your nucleus and helicase. This is all constant. You only change this. This you design in such a way that you see it is GACCG. So I have designed in such a way that it will bind to this part CT. So basically it will replace this part and it will bind this part. You see, I guess you all know the complementarity, right? A will bind to T, G will bind to C. That's what we were saying. So if I have this sequence, I design a sequence which is complementary, and this will go and bind to that. Now once this binds to that, that means it is actually binding to this region. And your Cas9 will be employed, or basically there is also Cas9, and then that can cleave off that region, and basically that will remove that part of DNA from your genome, right? So that will remove that part of DNA from the genome, and that you can use it for other things. You can, like, basically if you want to insert a new DNA into that, you can do that. If you want to have a different kind of small fragments, you can do that. But all of this, only thing you need to, now if I have to do a different sequence, I just have to change this sequence, right? This is a very, very small RNA. You can ask, how do we get this? This is nucleotides, right? These are probably around 25-30 bases. It is very easy to synthesize these 30 bases chemically. You synthesize that, and this will be the region that you keep on varying it. You keep, depending on where you want to bind this, you vary this part, all this will remain constant, and that part it cleaves off, and it's up to you what you want to do after that gets cleaved off. You can put a new insertion into that, or you want to separate it out. It's up to you on what are you ultimately aiming for. Okay, this is how this CRISPR-Cas9, which was a defense mechanism, is being used as a molecular scissor. Okay, yes.
So here the CGG is the codon that we wish to remove.
Right.
What happens to the other hydrogen bonds present in the Cas9 enzyme DNA?
You mean these hydrogen bonds?
Yes, sir.
These ones? These are not anyhow complementary. They will not bind.
The ones below the scissor, fewer.
These ones. Those will not be touched, only because it only binds, because this is the part of your single guide RNA which is bound to that, right?
Yes,
so this will not be cleaved. Only the DNA from the where you want to cleave, only those parts will be cleaved off. This will not be cleaved because it is a part of that. So there is again enzymes will have a certain conformation. So this will not fit into that. Only these regions are cleaved off. Yes.
How do we go about editing?
Right. So if you have cleaved off, right now, like you have, suppose I want to, it's easy. So suppose I want to remove a specific region. Now I want to remove this region. I have cleaved here, and I have I designed one more thing where I cleave this part. So this part is gone. Now this is hanging, right? You can do enzymes called ligases. Ligases means enzymes that, so we are talking about nucleases which break the phosphodiester bond. Then there are enzymes which make the phosphodiester bond. They will ligate that, and so you have removed that particular part. Or if you want to add a specific more kind of bases here, you can also do that by a reaction. I think some of you might know something called polymerase chain reaction, PCR, where you can add specific bases. So if you want to add specific bases here, you could do that. If you want to remove, you can remove that. So any editing first will start with cutting. After you cut, you can insert something or you can cut it other place so that you remove everything. So the first step is cutting, and after this it's up to you what you want to do. In the sense, you can add more bases by PCR, or you design one more Cas9 so that it cleaves off here so that this part is completely gone, and then you ligate these two by a phosphodiester bond. Okay, so that part. So as I told, suppose there are 3 amino acids that are creating a problem for me, that is resulting in a lot of diseases, right? I want to remove those 3 amino acids at the gene level itself. 3 amino acids means I have to remove 9 bases, right? Now I look at the genome sequence, I'll see, I'll cut here and then 9 bases after that, I'll cut that. So that part will go away. Then I ligate these two. So when my protein is expressed, it will express without those 3 amino acids. So it will not result any diseases. That's how it works. Yes.
So this mutation occurs during the birth of a child or when the DNA is synthesized. How can we add this Cas9 DNA?
Okay, so you're going into the cloning level at a much higher level. So, okay, so this can be done at an embryo stage itself, where even before the birth of a child, you could modify the genome so that the child will be born without any defects in the future, because that part you have removed. But as an adult human being, because already that protein is there, then you cannot do much. This has to be done at a gene level, right? So if I inherited disease, what is inherited disease? Inherited diseases are you are getting it from the birth from your parents, right? So you want to remove a specific disease, you have to do it at an embryo level itself, where the proteins are not being formed. Once the protein is formed, but also if the proteins are formed, right? It's not like protein, once it is synthesized, it will stay forever. It also has a certain lifetime, right? So if it is not very late, the proteins go back and you have a new protein synthesized. So if you can edit that, then the new protein that is synthesized will not have this mutation. Okay.
But sir, how do we know the DNA sequence that an embryo has?
You can do the sequencing. That is called human genome sequence. You can extract the DNA and then you can sequence the entire genome. So I don't know if you are aware. In the Western countries, they regularly do, but here also I think quite. Suppose a child, people do sequencing, not genome sequencing. It is still a genome sequencing to understand what kind of hereditary diseases the child might have, whether he'll be autistic. In India, people generally don't do that, but if you go to the Western countries, they do a profiling. Okay, what are the diseases that the child will be prone to? Right? What are the diseases that are basically, whether is he autistic or he's having any specific diseases that he's prone to. So those kind of things people do, where they sequence the gene of the embryo before they are born.
So can this system also be used for inactive cancer mutations in cells?
So this is a general technique. Anything that you want to edit at a gene level can be done, because as I said, so this is at a molecular level. As I said, everything, the root cause is your DNA. Now, this technique is allowing you to edit your DNA. So, in principle, like cancer, diabetes, blood pressure, everything has some genetic relevance to that. Now, like there can be a blood pressure because there is accumulation of cholesterol. That you cannot do because that has nothing to do with the genes, right? That is something that is of your habits that you have developed. But something that goes directly from the genetic inherit, that anything can be kind of edited. So at a genetic level, any disease or not only disease, suppose I want to have somebody who has, you all probably know that a part of intelligence is also kind of a genetic, right? So if you want to have a super intelligent person, you could also have that particular genes, right? But although it's a little controversial, but still it can be there, right? So everything is possible. Anything that can be, where you can edit the DNA. Yes.
Sir, how was this CRISPR-Cas9 system introduced into the body?
Oh, okay. So there are multiple methods like where you can introduce this biomolecules into the system. There's something called transfection. That is a higher level. There's something called transfection. If it is a bacteria, you could do something called transformation for a smaller, introducing plasmid DNA, other kind of. But generally, people do what is called transfection, other kind of thing, where you can introduce biomolecules into a higher living systems. Okay, that's a separate topic, but yeah, you can introduce that.
So
if we are introducing this into an organism, and the gene would be expressed in every cell, right? So how can introducing this?
That is why you have to do it at an embryo level. You have to do it at, I don't know, like you have to do it when it is a plant, not as a tree, right? At a seed level, you have to do. because then it is impossible that how will you go to every cell and do it. You have to do when it is a seed, because everything else is coming from the seed. So you have to do it at a very, you can't, as I said, at a much later level, it becomes, there is no point in that, right?
So in that case, you said it can be used to treat some genetic level cancer and also. In that case, if it's like, how can we detect it at such an early level? Like even if, when you're an adult.
Again, that's why I said this is for inherited diseases. Inherited diseases means there are certain mutations which cause this is not for diseases that come at a later stage. There are, as I said, there is some programming already, not complete. There is a part of a human body which is programmed. You know that this guy will get a guy or girl or somebody will get a certain disease. Nobody does the sequencing because you don't want to know what kind of diseases you get, right? So inherited diseases, okay, not for diseases that are because of lifestyle changes.
Sir, the exact place where we want to do the cleavage is specified by the guide molecule. Right?
Guide molecule, yes, the guide RNA. That is called single guide RNA. Okay, yes.
So now if a parent has cancer and the child at the embryo stage may have chances of getting the gene that causes the cancer mutation. So after doing gene sequencing, can we at that stage itself change the code?
That is little, I think it should be possible. As I said, anything that can, yeah, it should be possible when you can edit it at a genetic level. So it should be possible. Right? Yes. Sorry?
Is this
Okay. So I'll come to that. Now there are two things. Anything that is in science, there is always ethics involved. Okay. So you can develop a wonderful technology, but people will misuse it. This has already been misused to create 2 designer babies in China. Designer babies in the sense you have already designed how the baby should look like. And it is a big, there was a case that went on long back. So this has been used to have a designer babies. So always ethics come into that, in the sense like science can do a certain things, but there are something called ethics. Do you really want to eradicate all the diseases? Do you really want to, so should it be used or only for a certain thing? Because just like a nuclear energy, right? It can be both, it can be used as a bomb, it can be used to create your energy. Now once people have started using to have a specific, a character, designer babies or a specific thing, then people don't want to use it for those things, right? So most of this, there are companies based on this technology, CRISPR-Cas9, for eradicating diseases like diabetes and those. There are companies in the US, also in India. If you have looked at, there is some company called CRISPR Therapeutics, which works on using this technology to kind of work on the inherited diseases. Okay. Also, this particular technology was used to detect the COVID-19 virus actually, because it has a specific binding sequence. Yes. So to give an answer short, yes, I don't know if I have the slides for that, but there are a lot of companies that are working on using this technology for various platforms, and diseases is one of them. You could have a multiple things for that. You can like I don't see. These are all the things that people have been working on, as I said, to improve the quality of rice, to improve the quality of wheat, tomato, and this is all for the crop improvement. Then in human therapeutics, there are a lot of companies which are working on all this cancer. Then you have people working on AIDS, cystic fibrosis, and all this. There are a lot, because they know what gene causes what disease. So there are a lot of companies that are working on, and one of the companies, as I told, is CRISPR Therapeutics, which is very, very involved in using this technology to kind of treat or not diagnose, to kind of get away from this particular inherited diseases. But again, there are people who have misused it. As I said, there are in China, there are people who used it to, I think the story goes like this. They have taken the gene from HIV parents, both mother and father, and they have edited the gene which caused HIV, and they have introduced it into a kid to see whether the kid will ever get HIV or not. So it's like using that as a model organism, which is against the ethics. So people, basically people have done that, for which they have been jailed and other things. But yes, it has been both used positively and negatively. This is why I said this is such a revolutionary technology, is because one technology is able to it can be used for, as I said, it can be used for all these things like, crop production, it can be used for health, it can be used for, as I said, one project of CRISPR-Cas9 is to revive woolly mammoth. I guess you all know what is woolly mammoth. It is an extinct animal, ancestor of elephant, right, which is much bigger. So one of the project of CRISPR-Cas9, or one team is working to revive woolly mammoth. So if I have given you the science behind it, you can go back and see how CRISPR-Cas9 is being used these days on various projects, including crops, diseases, and people are using it for fun in the sense to revive some old extinct animals, and also people are using it to eradicate infectious diseases in mosquitoes and all those things. So this is basically, I would say, they want to have, these are all the applications. As I said, the unusual applications. As I said, you give a technology to humans, they'll use it for all possible purposes. They want to use it positively, negatively. There are people who are using to, like for example, like a pet breeding they are using, where they are making genetic mutations in dogs, especially Dalmatians, where this they have created Dalmatians without this specific mutation that will have a problem of bladder stones. Okay, and I think one interesting one is this one, de-extinction. Yeah, de-extinction is something that I said. So this Ben Novak's group, they have basically trying to extract the genes from extinct passenger pigeons, and they're trying to revive it. Okay, and then there are people who are using, I think some people are using to develop a much better horses for the races. Okay, because those are all having a shorter lifespan, it is easy to look into that. So I gave you some groups that are working on this. You can get a lot of other information from this particular website. Okay, it has been that is why I said 2011 it started 2015 the paper came, by 2020 the Nobel Prize came, because it is not that easy to get a Nobel Prize in like 5-6 years of work, because people are using it extensively for all the purposes, right? And so like you can, as I said, I think one thing I wanted to tell, yeah, even greener fuels they are trying to generate. Okay, faster, this is the one I said, faster race horses they are trying to generate. Because everything, any living species is a DNA, right? You edit the DNA, you can modify that. So, okay, so at the end of the day, people are thinking like, can the CRISPR be used as a promise of having a reduced hunger and inherited disease-free world? This is why if we can use CRISPR to reduce the hunger by improving the crop productivity and by kind of reducing the inherited diseases, this is the ultimate aim of CRISPR. Remaining all are kind of side things which are for but the ultimate aim of humanity is to have these two, and hopefully CRISPR will one day do that. So I suggest you to go back, read about CRISPR, you know how it works, the molecular mechanisms behind it, how things work. So you should go back to see what it is being used for. That is why this is called as a revolutionary technology. Okay, yeah, this is where I end my lecture, 5:30. Yes. Yes.
Since the CRISPR-Cas
Mic.
So since the CRISPR-Cas genes are basically from virus and bacteria, would the human body think that it is foreign and would it attack on it?
Good question. So the Cas9 genes, so the guide RNA is one that you are synthesizing based on where you want to attack. That is some. But the DNA, for example, Cas9 is a bacterial protein, right? Like there are quite a lot of bacterial proteins that we inherit. So people have looked into it, and there will be some immune response, but it is not huge. But yes, you're right that probably it will think as a foreign DNA and it will start attacking. But these have been probed, and I don't think there is a lot of immune response from our bodies. And especially you are going to do it at a much earlier stage, where like if you are working on embryo stage, there is no immune response there. There is only DNA, and you are editing the DNA, right? So it's not like you are doing on an adult human being having a huge immune response.
So in this CRISPR-Cas9 thing, when we try to eradicate pests and do things like making it a disease-free world, and also like modify things, bring back the extinct things, are we not going against the natural selection?
That's what I told you. Science doesn't care. Science is giving you technology. It's up to you how to use it. You use it ethically. I told you, Like there are designer babies. I don't know if you are born before 2000. I'm sure none of you were born before 2000. There was a sheep that was cloned called Dolly, completely using cloning techniques. Right now, it's up to you. The science will, as I said, it's like a nuclear bomb. I have shown how the nuclear fission or fusion works. Whether you make a bomb out of it or you make energy out of it is up to you. Right? Yes, there are enough instances where people have gone across the ethics and done that. But that is why there are ethical committees which do not permit the use of this for those kind of things. But in principle, you can do it is against the humanity. Yes.
Even when it comes to like correcting things, which is for our favor, isn't it like science going against science?
Yes. So that again, now you might say. So one of the ways of reducing malaria or dengue is to have mosquitoes reduce their reproduction. Now, is it ethical or not? So, in the sense like you don't want to get dengue or malaria. So, one way is to reduce the reproduction of your mosquito. But for mosquito, it is a living organism. Reproduction is one of its basic essential things. Right? Now, it depends on which area, what is the concern and other things. It is the same like, there are wild boars eating your farm. You want to kill the wild boars. Is it ethical to kill a wild boar? Because it is not. But you are destroying your crop. So, there are always ethical reasons, ethical committees which kind of do that. There is always a balance between what you want to do. The human science and technology is always in the sense that you can't balance both at some point, or you have to balance both, because otherwise it will be kind of, yes.
So when you're using the CRISPR-Cas system and you're taking a certain section of the DNA out, essentially the DNA, when it combines again, you're creating something new. What are the odds that this is actually good? What if it creates something unintended?
Again, those are mutations, right? So anything unintended is a mutation.
So what's the point of having the system?
But the chances of mutation are very low. You have cleaved off something and then you have ligated it. Any system, any living system or any system can go wrong sometimes. But what is the probability of it going wrong versus like it is similar to the example of going by a train can cause an accident. But what is the probability of going by that you always look at? Yes, it will cause mutations, but the probability is low. And you should also look at whether that mutation is probably more serious than the original one. Right? That depends on every case. That is a case study for every kind of thing. You cannot generalize that, but yeah. Most of the times there is no mutations, but there can be mutations, but you need to look into what exactly the system is.
Sir, for example, a particular person's DNA has been collected and he is free of that particular disease. Is there any possibility that the offspring of that person can have the disease? Or?
It should not, because your offspring is going to get a DNA directly from your parents, right? So the chances are very, very low.
But is there any possibility at all?
The other person, right? So there are two DNA chromosomes come from your mother, father. If your father corrects and your mother doesn't correct, and there is always a chance of that. So those are all questions that are still possible.
Sir, is there any way we just use PCR without cleaving anything?
Without?
Cleaving anything.
But how does, so DNA, if you want to introduce it, like this is my DNA, I want to introduce something here, right? Unless I break this and this, how am I going to introduce something?
What if at the end?
At the end you can do it, but at the end may not be useful, because this is where, that is what the endo and exo, right? So you have amino acid sequences here in the middle that you want to change or introduce. At the end you do, but what is the chance that all the amino acids that are important are only at the end? Most of them, because again it's a probabilistic event, right? Things at the end are very low compared to things. These are like 400, 500 amino acids. So the probability is 1 by 400 that particular amino acid is useful. So that is why you need to cleave to add something and then you join it.
So this is only editing the DNA within the chromatin and not we can't edit the chromatin itself, or?
So you edit it at a gene level. Gene becomes chromatin, right? So it doesn't become the organization. DNA is organized as genes, the first slide. Gene is organized as chromatins. Chromatins are organized, or they are all called genomes. So you do it at here, then this gets propagated. Like if you, it's like building a house, you kind of want to change like a specific not a good example, but I think if you want to do it at a macro scale level, if you do it at a micro scale level, that gets propagated to the macro scale level.
So that means, then can it be used for like Down syndrome and diseases like
Yes,
that?
the people have been using it. As I said, in the talk I could not cover, like it is impossible for me to cover all the possible diseases because I want to tell you the technique behind it. You go back to this slide or you even Google CRISPR-Cas9, you will see how extensively it is being used. The science remains the same because I only talked to you about science. Applications are multiple. Yeah, Down syndrome, yes. Any inherited disease, yes.
Or when this CRISPR-Cas9 was proposed by the scientists, was it proposed theoretically or did they actually experimentally try it out?
Yeah, the first experiment. So basically, I remember genome editing was going on. They got the sequence. They didn't know what to do, and they tried to look at. There are other genome editing methods that I didn't talk about, of which this is the most useful one. So they looked at that literature where this particular thing is being used as a defense mechanism, right? So initially, this was never proposed as a gene editing thing. This was more being understood as a defense mechanism. But then people use the defense mechanisms, the process of defense mechanism to edit the DNA, because just like you need to get the thought that okay, if I remove this, because I have said 100 times that genome editing, then you thought okay, if I remove this, it will go there. But people would not have thought that okay, if I remove this, it will take me to that and I can do a genome editing, right? That thought came to the Emmanuel Charpentier and Jennifer Doudna, and that is what led to this.
But is it legal to do genome editing at that time?
Yes, it is legal.
It is?
It is legal. A lot of, as I told you, there are GM crops, genetically modified crops. What do? Genetically modified crops have a modified DNA, genes, right? So it is legal.
Sir, you told about reducing the reproduction of mosquitoes. So, if there is any specific trait that you have, it is going to be carried to the next generation through reproduction. But if you are going to reduce reproduction itself, then how?
That is why, that is how they are going to plan. One of the plans to, this is an example. If you have a lot of stray dogs in your, the general example, what do municipal corporation do? They neuter. What do you mean by neutering? They are removing their ability to reproduce. Now, it is basically you have to decide, I will not have future dogs, or should I save myself from the dog bite, right? Yes, you are not going to have a next generation at all, but that is probably needed. Like if you see all the stray dogs, all these municipal corporations, they never cull them, they never kill them or cull them. What they do is they neuter them to stop the production. The same principle is being applied for the mosquitoes to stop that. Okay? Yes.
Sir, can a chromosome be added or taken away, like Down syndrome, Turner syndrome, for example, where there's a missing chromosome? How would it be done in that case?
That's a good question, but for that you probably do, if the entire chromosome is missing. A chromosome has a lot of bases, as we saw, right? So that is, you can edit a specific region, but if you want to entirely add that, we are talking about editing only. It goes there, cuts that. If you are basically have to bring in it, then this technology is not probably correct for that. There are other technologies maybe, but this technology cannot do that because this is only about editing a specific sequence of DNA, right? You can't add, you can, within the edited sequence, you can introduce new DNA, but you can't bring in the entire gene to do that. Okay, so shall we stop here? Okay, thank you.
Sir, thank you for this wonderful session, sir. It was a nice experience, and I think everyone understood the concepts.
My aim of this lecture is not to give you advanced science, of course advanced science, but I wanted to teach you the basics, how you don't have a difference between various things, how biology is nothing but chemistry, but also how one particular thing can be correlated to other, and then how it can go to application. And for that, this is one of the best examples. Thank you. I'm glad that you all enjoyed.
Thank you for sharing your knowledge.